last class we have discussed about the digestive enzymes. Okay, uh, I think you have gone briefly for that. So in that we have talked about the digestive enzymes, different types of digestive enzymes, and their role. Okay, where they are secreted. These enzymes I have already told they will be dealt separately in the particular digestion, like protein, carbohydrate, lipids separately, and also we have talked about the zymogens. Okay. Zymogen types of enzymes means as a proenzymes. So why they are synthesized as zymogens? Some of the zymogens enzymes like pepsinogen, trypsinogen, chymotrypsinogen, proelastres, prophospholipase, all these pro type enzymes, they are zymogen, means they are need to be activated when they are being secreted in the particular sites. Okay. So for activation, then the certain part of that protein will be clipped, means their active site will get exposed. Okay, so these things we have learned in previous topic, and we have talked about this secretagogy, means the certain molecules that are helping for the secretions of other enzymes or substances that may include hormones and neurotransmitters. Okay, acetylcholine, serotonin, and other hormones like somatostatins, gastrin. These are the secretagogy. Now today we are talking with the pancreatic secretions. Okay, so pancreatic gland, they may be acting both as both endocrine and exocrine glands. Okay, endocrine glands, as a function, they may produce the different types of hormones, insulin, glucagon, somatostatin, and after that, pancreas secretion contains the pancreatic juice. Okay, and these juice are in the small intestine, means they are secreted in the small intestine by a dog, and the role is to neutralize the pH and become suitable for the action of pancreatic digestive enzyme. Okay, the pancreatic secretions containing the pancreatic juice, helps to neutralize the pH of that the food coming towards the small intestine, because the food which entering into the stomach, or we can say gastric gland, so always they are exposed with the HCl, means highly acidic environment. There is highly acidic environment. Now, so the acidic food should be to some extent neutralized. Okay, by the, and that is being carried out by pancreatic secretions to neutralize those acidic phase substances, and mainly it is of that bicarbonates. Okay, so for the complete digestion of food requires both pancreatic and cross-border enzymes. Most pancreatic enzymes are produced as zymogens. We have already discussed. They are required to produce as zymogens because there may be the chance of that self-digestion. Okay, auto-digestion process means the digesting the same cell or tissue which are being produced. And trypsin. Okay, trypsinogen is the main activator molecule. Or first, it should get converted to trypsin by the enterokinase, and that trypsin can help for conversion of different other coenzymes. Or the different proenzymes for their active form. First trypsinogen to trypsin, and it has capacity to convert other coenzymes to its active form. And there is also the in pancreatic cells or tissue, we can say there is pancreatic trypsin inhibitor. It means it inhibits the erroneous activation of trypsinogen. If by false, by error, if trypsinogen get activated, then autolysis of that cells in them, cells in tissue, so that by chance or by error, if they are being activated, then this inhibitor helps to inhibit the erroneous activation of that enzyme trypsinogen. So trypsinogen is converted to trypsin by enterokinase, and this trypsin helps for activation of inactive enzymes that are zymogens to active enzymes. Right? That is the main role. Pancreatic juice. So pancreatic juice, talking about pancreatic juice, contains water, bicarbonate, and digestive enzymes. Okay. water. The main content of pancreatic juice are water, bicarbonate, and digestive enzymes. Enzyme, you can see the list of enzymes. Many enzymes are present in this pancreatic juice. Trypsin, chymotrypsin, elastase, carboxypeptidase, lipase, amylase. Some enzymes are responsible for the digestion of carbohydrate and some for lipid and proteins. Okay. And they have different actions, cleaving peptide bonds or fatty removal of fatty acid from glycerols like that. So we can say the pancreatic juice measure, it contains water, bicarbonate, and digestive enzyme. So bicarbonate is also important component of pancreatic juice. I have already told major function is to neutralize the acidic in acidic containing food because they have been exposed in the hydrochloric acid of that stomach. And when it comes towards this small intestine, so highly acidic environment, they should get to some extent get neutralized by this bicarbonate. That was about pancreatic juice. After that, bile juice. Okay. So bile juice is also helping for the digestion and absorption of food materials. And they are dark to yellowish brown fluid that is produced by liver, stored in the gallbladder, and are discharged into duodenum. Right. So bile juice also detailing you will be learning, but you have to know certain fundamental things of this bile juice being stored in the gallbladder and components. When you talk about the components of this bile juice, there are water, bile salts, okay, bile pigments, and cholesterol. So, excretory route for cholesterol is that through the formation of these bile salts. And bile salts function because bile juice has bile salts, they act as emulsifying agents in digestion and absorption of fat. Mainly, we can say bile salts has a role in the digestion and absorption of fat. So, and these cholesterol bile pigments are broken of hemoglobin and excreted from the body in the bile. So, bile pigments are also removed, and cholesterol are also removed for the formation of this bile acids or salt, you can say. Their root. Okay. So, bile juice, an important component of bile juice is bile salts. Bile composition, when you see the bile juice composition, there is water, obviously bile salts, right? Chenodeoxycholic, deoxycholic, lithocholic acids. Right? Means these are the acids, and when they are they combine with this sodium and potassium salt, they form the salt. Okay, this deoxycholic acid, lithocholic acid, and there is mucus in this bile juice. Pigments, bile pigments are generally bilirubin, biliverdin are bile pigments. Such type of pigments are also found in bile juice. Fats, 1%, phospholipids, lecithin, cholesterol. Okay, so these are the composition of bile juice. Gastric secretions. Now coming towards the composition and function of gastric secretions. So we have learned about the pancreatic secretions. HCl converts pepsinogen to pepsin for chemical digestion. HCl itself is a strong acid, so it helps for the digestion to break into each other components. After that, activation of pepsinogen. Optimal pH environment for pepsin, because pepsin requires highly acidic environment. The optimum pH is 1 to 2. And that environment is created by this HCl in that we can say stomach lining, stomach environment. Another function, it destroys the bacteria. Promotes the absorption of calcium, iron in the stomach. Other composition of gastric secretions. Pepsinogen. So pepsinogen is also the digestive enzyme, right? Coenzyme, and because of pepsin. And after being activated by HCl, convert to pepsin and a role for the digestion of proteins. Another, there is mucus. Okay, it forms a protective barrier. Mucus protective barrier, it forms through the mucus. After that, intrinsic factor. Okay, there is intrinsic factor, and that factor is required for the vitamin B12 absorption. Okay, only when the vitamin B12 is present in the GI tract, after combining with this intrinsic factor IF, then only it is be able to be absorbed. Okay, so proper absorption of this vitamin B12 it requires intrinsic factor, and that is the composition of gastric secretion. So in gastric secretion, we can say there is HCl, pepsinogen, mucus, intrinsic factor, okay, as a major components. Now that was about the different secretions, GI secretions, gastric secretions, pancreatic secretions, the bile secretions, and their composition as well as their function. What are the functions carried out by different secretions? What are the importance of those secretions in digestion and absorption? Okay. Now coming to sodium absorption. Okay, sodium electrolytes absorption. So 20 to 30 grams of sodium are secreted in the intestinal secretion each day. And a person, we only eats generally 5 to 8 grams of sodium each day. So to prevent the next loss of sodium into the feces, the intestine must absorb generally 25 to 35 grams of sodium each day, which is equals to the 1/7 of all the sodium present in the body. That is, in our body, the total amount of sodium that is being present in the body, about that 1/7 need to be reabsorbed or absorbed through the GI tract. Okay. And how the sodium get absorbed? So the main for the absorption of sodium, it requires active transport of sodium. Okay. For active transport of sodium is from the inside of the epithelial cells. And the side walls of these cells into paracellular spaces. For the sodium should be excreted into paracellular spaces and towards the basal sites. Okay, and that is the active process. The sodium potassium form is helping for that. Okay? 3 sodium out and 2 potassium in. First, the active transport is there in the basal surface, basal site. Okay. Now that because of that active transport, the concentration in the epithelial cells inside the cell is about generally 50 milligram per liter. It will be reduced to low concentration. Because of the sodium concentration in the chyme, or you can say basolateral site, the content is 142 milligram per liter, and this causes the electrochemical gradient. Okay, and so it helps for the more sodium ions to be transported by the epithelial cells into paracellular spaces. First, for the absorption of sodium, because it is anions, cations, we can say sodium potassium active transport first, basolateral site. Okay, active transport causes the through the figure. We can go through the figure. Okay. First, apical site and basolateral site. First, what is happening is that through the active transport process, the sodium is being pumped to the surface. Okay, sodium potassium ATPase. Now, when it is being pumped out here, space the inside the content of this epithelial cells lining will decrease to 50 milliequivalent per liter. Okay, so here the sodium may be transported through the symporter, sodium glucose symporter, or through this diffusion process. It may get because of this electrochemical gradient. And because here it is being pumped out by the sodium potassium ATPase this side, and now due to electrochemical gradient will be created, and because it reduces the content of sodium up to 50 milliequivalent per liter, and because of high sodium this side, the gradient causes the diffusion of this sodium in the up to here. Okay. From epithelial lining cells, or from here to into here, yeah, there should be active transport. Okay. And another thing, for the absorption, when sodium are being absorbed from here to here, the water also follows the direction. Okay, water follows salt, water follows sodium through the epithelial. So because of this osmosis, first sodium is being transported. Sodium is being transported through the across this membrane, okay, through the diffusion, and the consequence is that the water molecules is also following the sodium. Okay, so we can say water follows the sodium through the epithelial membrane. Because of this electrochemical gradient, sodium is being transported. Because of that, osmotic gradient is created, and water is also flowing the direction of that, okay, causing the flow of water from this side to that side. Absorption of chloride ions in the duodenum and jejunum. Generally, sodium also are absorbed in this duodenum and jejunum parts. Now, with that, chloride ion also being absorbed, because in the absorption of sodium ions, so the epithelium creates electronegativity in the chyme, electropositivity in the paracellular spaces within epithelial cells. And then chloride ions move along this electrical gradient to follow the sodium ion. Because of flowing of that sodium ion, first, sodium ions are being absorbed in epithelial cells and towards the chymal site. And because of that absorption of sodium, it is creating the electronegativity in the in the kind of electropositivity in the paracellular spaces between the epithelial cells. Right? Because of this gradient, now the chloride ions move along this gradient to follow the sodium ions. Yes? Chloride is also following that sodium and hence get absorbed. So this was for the absorption of sodium. After that, chloride is also following the sodium and water through the osmotic gradient, osmosis. Coming to cystic fibrosis, okay, disease called cystic fibrosis. And this type of disease in cystic fibrosis, the mainly affected organ are lungs. The pancreatic insufficiency is there. Pancreas is also being affected. When the lungs are being affected, it causes difficulty in breathing. Pancreatic insufficiency is there. Lack of digestive and secretory pancreas. There is intestinal blockage. Lots of chloride in sweat. Male stereotype. So in cystic fibrosis, these are the complications that we may find in lungs, pancreas. So because what is happening in cystic fibrosis is that there is defect in chloride transport. Okay. So chloride transport is being defected, and these are the affected organs: lungs, pancreas, lots of chloride in the sweat, and also it may be responsible for male stereotype. Cystic fibrosis is prevalent and serious genetic disease and affecting approximately 2,500 individuals among the 2,500 populations inherited as autosomal recessive disease, and about 1% in 25% is a carrier. Okay, this is about the cystic fibrosis. It is the disease of exocrine glands. With the respiratory and GI tracts being most affected. And the diagnostic hallmark is the presence of high amounts of NaCl in sweat. So there is the reflection that there is abnormality in the exocrine gland function. Okay. This is about the cystic fibrosis. Genetic disease, highly serious genetic disease among the North Americans. Occurrence is 1 in 2,500 individuals, inherited as autosomal recessive disease. And the diagnostic hallmark is the presence of high NaCl, amount of NaCl in sweat. So in cystic fibrosis, there is high amount of NaCl in sweat. Okay. Sodium chloride is excreted more. So to understand the cystic fibrosis, we should understand the receptor. Okay. For this chloride channel. First, about this epithelial sodium channel in a CFTR, cystic fibrosis transmembrane receptor, regulator protein. So this is for chloride channel. Okay. We can say for chloride secretion from this side to this side. So you can see vasculator apical side. Okay. And this CFTR is the main chloride channel in epithelial of various tissue. Generally lungs, we can say. Sweat gland, we can say. Pancreas, we can say. Right? So these are the location of the CFTR receptor for chloride channels. With the absorption of sodium and chloride from basal side, it follows chloride also for this side. For the in the apical side, there is epithelial sodium channel, and also for chloride there is different CFTR. Okay, for water there are aquaporins. Secretion of NaCl and water in normal epithelial cells. So same thing, how they are being transported. First, sodium is being pumped. Okay, for sodium, it follows the chloride as well as water. And this apical side, there are sodium channels, chloride channels, okay, aquaporins for water, and water follows the salt because water salt sodium is being followed. After that, due to electrochemical gradient, chloride also followed. Because of osmotic gradient, water also follows here. Okay, up to here, and after that, they are being transported by different transporter protein, also aquaporins. Some characteristic features of CFTR protein. Okay. They are located in the genome chromosome number 7, 25 exons, transmembrane protein of 14 and 18. There is 2 nucleotide binding F domain and regulatory domain. This type of CFTR channel has 2 NBF, okay, nucleotide binding domains and 1 regulatory domain. So because of this cystic fibrosis, in cystic fibrosis, mutation in there. Mutation because of deletion in this 508 position. The phenylalanine is being removed. Okay, because of that mutation, NBF1. So we'll talk about that. What is happening in cystic fibrosis? So this is CFTR protein. Okay, NBF1 regulatory domain, NBF2 nucleotide binding domain. Okay, nucleotide binding domain 2. Second, so in cystic fibrosis, the NBF1 domain is mainly affected. So what is happening here in this cystic fibrosis? The mutation is present in 508 position. French mutation is there, means one amino acid will be removed. That is phenylalanine. Other mutations are also there. That is the most common mutation. Right? Most common mutation is the deletion of this 508 position amino acid. That is phenylalanine. That will be removed in this NBF1. Okay, and that is the genetic basis for cystic fibrosis. Okay, obviously other mutations are also there, but this is the commonest type of mutation that we found in cystic fibrosis disease. Okay, CFTR is a cyclic AMP responsive chloride transporter. Okay, means the mediator is cyclic AMP. Bicarbonate ion production in pancreas. Okay. So I have already told in the pancreatic secretion, the bicarbonate is also the important component. Okay. So how they are produced in the pancreas, how they are produced and secreted. Okay. First, for the production, the CO2 is diffused into the ductal cells from to the interior of ductal cells from blood. First, there is the diffusion of carbon dioxide from blood, and that combines with water, bicarbonic anhydride to form H2CO3, and that is again dissociated to form bicarbonate and H+. And bicarbonate is actively transported into the lumen. First, absorption of CO2 from the blood, combining with water, it forms bicarbonate, carbonic acid, H2CO3, and that dissociates to form bicarbonate and H+. The bicarbonate is quickly transported into the lumen. Okay. The H+ formed from the dissociated H2SO is exchanged for sodium ions by active transport through the blood, which will diffuse or actively be transported into the lumen to neutralize the negative charge of bicarbonate. Okay. The movement of bicarbonate and sodium ions into the lumen causes the osmotic gradient, causes water to move from the blood to the ductal cells of the pancreas, producing eventually the bicarbonate solution. So this is the theory of production for bicarbonates. How can I be explaining through the bicarbonate ion production in pancreas? Okay. So this is the pancreatic lining cells, you can say. So one portion is being enlarged. This portion has been enlarged here and shown here. Okay. The cells, acinar cell is this, one acinar cell, and the blood lining is there. So there is diffusion of CO2 from the blood vessels to the acinar cells. Combining with water, it forms carbonic anhydrase, Ca. After that, this carbonic acid is formed, H2CO3, and that dissociated by carbonic anhydrase enzyme, bicarbonate and H+. Okay, bicarbonate is being secreted towards the lumen, or this lumen here, this one here, lumen. So with that, H+ is being expelled out through the by this mechanism, sodium proton pump. Right? This is sodium proton pump. Here you can see this figure. By formation of carbonic acid, splitting into bicarbonate and this proton H+, that is being exchanged with sodium now from here. Now sodium is being entered into these acinar cells. Along with that, water is being flowing. Okay, because we know water follows the salt. Because of that osmosis gradient, now water is also flowing through this when the sodium are being absorbed. Okay, and sodium comes to us this. This sodium also being follow this bicarbonate, water, sodium, and they are coming to us this lumen site, okay, of this pancreatic acinar cells towards the lumen. So in this way, in this lumen there is water, sodium, bicarbonates, okay, and in this dock. Now they come from this lumen, acinar lumen to dock, okay. So the pancreatic dock, and through the docks, now they can come into the GI tract for these secretions. Along with other digestive enzymes. Okay. First, this we are talking about the bicarbonate, how they are being flowing, taking the route. Okay. So in this way, bicarbonate ion are produced in the pancreas, and they are secreted. Okay. So that's why they are the important component of those pancreatic secretions. So in today's this class, you have learned about these different types of secretions. Okay, pancreatic secretions, bile secretions, gastric secretions, the content, their role, and also about the cystic fibrosis, about the disease cystic fibrosis, the defect in the chloride channel transporter. Okay, cystic fibrosis mutation is caused in that channel, CFTR protein, and that causes defect in the chloride channel. And the mainly affected organ are pancreas, we can say lungs, pancreas, and also other sweat glands, excessive NSM in the sweat glands. So if you have any problem or doubt, you can keep over this chat section. Okay, any message or in this digestive enzyme, we have talked about the digestive enzymes, their role, and other different secretions. Okay, like gastric secretion, pancreatic secretions, bile secretions, their role, and also about the chloride channel mutation in the chloride channel, how the bicarbonates are produced in the pancreas, how they are secreted. Okay, and about different neurotransmitters, secretors will be acting as a secretion of other different digestive enzymes and helping for the secretion of digestive enzymes and other molecules. Okay.